All right, good morning, everyone. I know that it is bright and early, and I am so proud of all of you who have signed on here at 8:30 in the morning. We'll be in Pear Deck today. This is our last instructional session before spring break. We'll come back together on April 14th after today for instructional sessions. But we'll be back in Pear Deck today, and so you see the code up there is HHFGK. And you can go to joinpd.com. I'll also put a link in the box, in the group chat box over here. Let me know. You can type in the chat room if you have any question while I'm presenting, or if the code disappears and you want me to type the code in. And if you were in another class, that is crazy because that's before 8:30, so I'm so proud of you. All right, let's get started. I'm going to start class. So we are in week 12 of our remote learning due to COVID-19. This is our second presentation this week. Just a quick reminder, make sure you're signing into Schoology every day and checking on all of your classes, and make sure you go to Google Classroom and check the stream and go to classwork, or sorry, it should say classwork, not classroom. You should go to classwork and check your work, look at your grades, and also check for any new assignments. I've been grading and trying to leave feedback for you so that if you would like to resubmit something, you can do so. Okay, so my question for you today is, what are you looking forward to the most during spring break? What are you looking forward to the most? You can draw anywhere on your slide. Welcome if you've just joined. We are in Pear Deck. Here's the code. The code is back up for you to see, is HHFGK. You can join using that code. Once again, I'm so proud of you guys for being on so early. Okay, I see some responses. I'll share them in about 30 seconds. Okay, let's see what we're seeing. So some of you say that you're excited about getting a break from classwork, as teachers are giving tons of homework, so we're glad to get some time to do it. I feel the same way about grading. But we want to make sure that you all are still learning and we're getting feedback, and that hopefully it'll get a little better after spring break. Someone's excited once again for not doing work. You've been working for the last 3 weeks and it's been a challenge. But you guys are rising to the occasion, and I'm super proud of all of you doing so. But yes, enjoy your spring break. Someone else says finishing all the work. And ooh, someone said working out more. Nice. And cooking more healthy foods. Yeah, I've been working out and trying to move more at home. And so I totally agree with you. Spending time to relax. Yes, I plan on reading lots of books. I'm excited for that. Someone said finally getting a break from all of the work. Yes, I feel like that is the sentiment for everyone. I think you guys are going to be just amazing scholars. And this is really going to get you ready for college, because you get a chance to see what it's like to be able to do a lot of your work on your own, which is amazing. Okay, well, I am looking forward to spring break as well. Let's jump into our lesson. So we'll have time for questions at the end. Okay, so what are we learning today? So today we're going to continue learning about how DNA is transcribed to RNA and how RNA is translated to create proteins. We're going to learn how do we read mRNA to create a protein. We're going to learn about the genetic code today. And we're going to learn this by taking notes on the presentation in Google Slides. You'll also, if you haven't done so already, you need to read and watch the videos and simulations in labexchange.org. And you're going to show you learned it by answering the questions in the assignment in labexchange.org, watching that ed puzzle if you haven't done so. And I forgot to add it here, but it's at the end. You have a DNA mutation challenge assignment. Okay, I just want to see if we remember from last time, what order would you arrange these in? So we have DNA, mRNA, protein, transcription, and translation. And on your screen, you can drag the numbers to where you believe they belong. So let's just this is just a quick review from last class. What is the order of these terms? Let's take about 30 seconds and determine the order of these terms. What comes first?
Dr. Haynes, I have some questions.
Okay, 1 second. I will, if you can type it in, that would be great. All right. Let me look at the responses. Let's see. Okay, so we have ones in a couple or a few places, but we do start off with number one, starting off here with DNA. So you start off with DNA, and then number two would be that you go to transcription. So I see one person who has number two here for transcription. And transcription produces mRNA, which will lead us to number three. mRNA is produced, and then mRNA goes through a process called translation, which would be number four. Good, I see four is over here. It's a process called translation, and then you end up making protein, which would be number five. And I see a five back here. So this does mean that we still need to review this. And I know someone said they had a question, so we'll make sure we have question time at the end as well. But I will be reviewing this, hopefully to make it a little more clear. Okay, so hopefully you can see this GIF. It's showing you the process of protein synthesis, which is what we just reviewed. So you start off with DNA, and the DNA gets made into mRNA via a protein called RNA polymerase. You should see that happening. And then that mRNA leaves the nucleus of the cell, and then it goes out into the cytoplasm, and through a process called translation, it makes amino acids, which makes a protein strand. And it does that with mRNA. And we'll be going through that process a little more in depth today. But just to show you a different visual, hopefully you can see here. So we start off here with DNA. And then DNA is double stranded and it gets opened. And one of the strands, called the coding strand, this strand is then used to make mRNA. That's what's happening here. And remember, it's the same binding principles where A's bind with T's, but because RNA does not have T's, it has U, it has a uracil. And anytime there's an A, you get a U. So it's the TAC makes AUG, and then TAG makes AUC here. And after that, that RNA then goes on to the cytoplasm. So transcription happens in the nucleus over here. And then the RNA leaves and goes to the cytoplasm. And this is where translation occurs. And so you get this translation happening in the cytoplasm where you make a polypeptide, which is another word for a string of amino acids to make a protein. All right, so let's look at translation just a bit more. So over here we have a ribosome. Ribosomes are what is important for making these polypeptides. So translation happens on a ribosome in the cytoplasm. So it occurs in the cytoplasm on the ribosome, just like you see there with the mRNA in the ribosome and the amino acid. I love these jokes, guys. All right, so let's look here. So there are 3 types of RNA involved in translation. So rRNA, this is a type of RNA that is part of a ribosome. This is ribosomal RNA and it's needed for translation. And then you have messenger RNA, which is the mRNA. Hopefully you see that there, okay. And this mRNA is the transcript of the DNA, which we've learned about. And then lastly, you have tRNA, which stands for transfer RNA. And this transfer RNA is what carries the amino acids to the mRNA so that this chain can be made. Okay, so in translation, mRNA, which you see here, is read in sets of 3 nucleotides. These 3 nucleotides are called codons. And I know that this is quite a bit of information, but this is being recorded and you can rewatch it and also ask me questions. But mRNA is read in 3 sets of nucleotides called codons. And so this right here, AUG, is called a codon. Anytime you have 3 nucleotides together on mRNA is a codon. And then tRNA, so this whole red thing here is called a tRNA. It brings the correct amino acid, which is up here. This is an amino acid called methionine, but we'll just call it MET. It brings the correct amino acid that matches the codon with this anticodon. So the tRNA has an anticodon. So the anticodon of AUG is UAC. It brings the right anticodon, and with it, it brings the right amino acid for that mRNA because that's the genetic code. So these are codons in this picture here of translation. UGG, AAA, GAU, and UUC. These are all codons. And then you can see the anticodons on the tRNA here. And so the anticodons bind to the codons on the mRNA right around the ribosome. And as they bind, they bring a amino acid and you start to make a peptide chain. And a peptide is another word for protein. So you're making a protein chain. Those are the amino acids. All right. Sorry. Here we go. So this looks, I know this slide looks crazy, but I'm going to teach you how to use it in the next slide. So these are 2 ways that you can read the genetic code. So this right here, this first box is the traditional way to read the genetic code. And the box, and this circle on the right is a newer way to read the genetic code. But one thing I want you to know about the genetic code and about proteins is that every single protein has to start off with the codon methionine. So you see a green circle here and a green circle here. This MET means start. If you don't have AUG as the first codon, the protein will not be made. It has to start off with AUG. And that's right here. AUG codes for MET always. And you can see AUG codes for MET. And then there are stop codons. So UAA, UAG, and UGA all mean stop. And you can see them here. They say ter for terminate on this side. And so anytime you see UAA or UAG, you have to stop. Doesn't matter if there's more codons after, you stop making a protein and you don't make it anymore. You just completely stop no matter what is following the stop codon. Okay, so I'm going to teach you how to read the genetic code for a specific mRNA strand. So this RNA strand has AUG, GCC, UGA. So the way we find out what AUG is over here. You read the first letter. So the first letter is A, which was just highlighted in yellow, hopefully for you. And then the second letter you read from the top, and that letter is U, which is now highlighted in yellow. And then the third letter you read from the right side. So now we have A, this row, U, this column, and then G right here, which is the 4th mini row. AUG will give you methionine or MET. So that's AUG. And then you do the same thing for GCC. Hopefully it shows up. Okay, so GCC, we have G here, C here, and then you have a C on the right side. GCC gives you alanine or ALA. And then lastly, we have UGA, which you look over here for the first letter is U. And then the second letter you read from the top, G. And then the third letter you read from the right, A, which means stop. So then you're going to stop making this protein. So this protein only has MET and ALA. Stop doesn't make anything. It just tells it to stop. So I want you to practice. So I want you to translate this mRNA strand by circling the correct amino acids. So I want you to circle the correct amino acids on your screen based off of what you see with this mRNA code. I just want us to see if we can read this genetic code chart. Remember, you start with the first letter on the left, second letter on the top, and the third letter on the right. So your first codon should be AUG, and you're going to circle what or identify in some way which amino acid you'll get for AUG. So I don't want you to give me the letters. I want you to identify the actual amino acids. So if it's methionine, circle methionine. If it's proline, circle proline. If it's glutamine, circle glutamine. I want you to tell me the 3 or 2 or how many amino acids you'll get from this mRNA strand. So I'm going to go around. Okay, I see somebody who has 1 and 2. Okay. Good. Close. Oh, okay. We have to like practice. Okay, I'm going to show one of your classmates' screens. Let's look. Okay, so the first thing we have to do is you have to separate these into 3s. So the first set of 3 is AUG. So I see this person did select AUG correctly. AUG is methionine, which means start. So we have A on the left side, U at the top, and then G from the right side gives you methionine. But then the next one is ACG. So once again, you go A on the left side, and then you do C from the top, and then G from the right. This ACG will give you threonine. But the next one is UAA. And this one is incorrect because it would have to be U from the left side, but then not U on the top is UAA. So it's U from the left side and then A, and then you have A again. So it would mean stop. So you would have methionine, threonine, and stop. And I'm actually going to stop here just for the sake of time, and then everything else I had planned will start after spring break. But I really want you to be able to answer, to ask me some questions, and I can go over what you see here. But great job. Let's discuss.